Hi everyone, my name is Lilith and I'm one of your IAs for BIBC 120 this quarter. And this is my first review video on Lecture 1 and 2. This will be a much shorter review video because Lecture 1 and 2 are just the beginning of this quarter. So let's go through it and you can expect for more review videos to come out as the quarter goes on. So in Lecture 1, we started to talk about what nutrition is and what nutrition science is. So you can think of nutrition as the consumption, absorption, excretion of food that we eat on a daily basis and how that is helping us carry out the processes that we do as humans. Nutritional science or nutrition science focuses specifically on how nutrition and medicine interact with one another. How does eating specific foods affect one's health? And nutrition science also looks at dietary and health issues. Now, there are two types of nutrition that scientists and medical providers can look at. So classic nutrition is describing the things that an organism needs to survive, right? So these are the essential nutrients that one needs to live. And if we take away certain nutrients, right, will this individual die, right? If the answer is yes, then we're talking about classical nutrition. Now, modern nutrition is different in that in modern nutrition, we are talking about things that the organism needs to live a better quality life, right? So not necessarily foods and nutrients that are essential to life, but nutrients that make our life a little better. For example, fiber, right? Fiber falls into the discussion of modern nutrition because without fiber, we will survive. However, with a fiber-rich diet, we will live a better life. In other words, you can think of classic nutrition as the study of nutritional deficiency, right? There are many diseases where, because of nutritional deficiency, there's an illness because of that. However, in modern nutrition, we focus on nutritional overabundance, right? Maybe eating too much of a certain food, let's say a certain fast food, is giving us a bad quality of life. So hopefully you can see the difference between classic and modern nutrition. And it's important to also understand that, a certain individual can experience both malnutrition while overeating. So these two worlds are interacting with one another and overlapping with one another. You can think of an individual who can eat or overeat a lot of fast food. However, that fast food is not fiber rich or nutrient rich, so the individual, although they are overeating, they will still be malnutritioned. Okay, then we started to talk about some of the essential nutrients. Essential nutrients, as the name suggests, are essential, right? They're needed to survive. So there are two types of essential nutrients: energy-yielding nutrients and non-energy-yielding nutrients. Energy-yielding nutrients are nutrients such as the carbohydrates, which are sugars. Here is, I think, glucose. I think this is what the structure of glucose is. Lipids, such as triacylglycerides, right here. And proteins. Here is a dipeptide for you, if you've taken biochemistry before. And all of these three nutrients or macromolecules are energy yielding, meaning that we are able to burn them and get ATP from them and be alive. The non-energy yielding nutrients are vitamins such as folate and minerals such as calcium and sodium and potassium and so many more. Now, these are nutrients that are still essential in our diet because we need folate to, for example, many pregnant individuals take folic acid, right? Folate, because it is important in genetic material building, I believe. And then minerals are important in your heart muscle, or sodium and potassium are important in electric transduction in your neurons, right? So these are still essential, however, they are not energy yielding. They will not give us ATP. Two other ways we can look at nutrients are through the lens of macronutrients and micronutrients. The macronutrients are nutrients that we need in large quantities. So usually the macronutrients are the energy-yielding nutrients, the carbohydrates, lipids, proteins, plus water, because although water is non-energy yielding, it is still considered a macronutrient because we need it in large quantities to survive. Micronutrients are nutrients that are needed in smaller amounts. Then we also talked about the non-essential nutrients, right? And non-essential nutrients are either phytochemicals or zoochemicals. Phytochemicals are found in plants, and zoochemicals are found in animals. And these nutrients, although they're good for us and they will benefit our health in some way or other forms, they are not essential to survive. Okay, an example of phytochemicals that we talked about in class was a cup of coffee, right? A cup of coffee can have about 1,000 different molecules of phytochemicals. And that was pretty much it for lecture 1, right? Just a general overview of what nutrition is and different types of essential and non-essential nutrients. Going into lecture 2, we started to talk about, well, we talked about these nutrients. What do these nutrients give us? And these nutrients give us energy, right? If they're energy yielding. And how is energy quantified? Energy is quantified through the word or unit of calorie, right? And calorie is the heat needed to raise the temperature of 1 gram of water by 1 Celsius. Now, kilocalorie, which is written like this, or with a big C, with a capital C, is the amount of heat needed to raise not just 1 gram of water, but 1,000 grams of water by 1 degree Celsius. So those are just 2 units that we will be covering throughout this quarter. Now, because we know that the foods that we eat give us energy, the Food and Nutrition Board had to come up with some sort of, you can call them, references or boundaries to help the public population figure out how much they should be eating of what kind of food. So the Food and Nutrition Board came up with these dietary reference intakes, or short DRIs. There are many DRIs. Over here, we have the estimated average requirements, the recommended dietary allowances, the tolerable upper intake levels. And all of these DRIs basically tell us, maybe you shouldn't be eating too much of this certain food, right, past the upper limit level, or the estimated requirement of a certain food that one needs to survive is decided by the EAR. So you can think of these as general guidelines for the public to follow to be able to live a healthy life through their dietary intake. Okay, for the exam and studying purposes, I would advise you to go through the definitions of each of these DRIs and just make sure you know the difference, and also keep this chart in handy in your notes. Okay. Now, it was great that the Food and Nutrition Board came up with these DRIs. However, imagine going to buy a protein bar, and now on the back of the protein bar, they have to fit all of these different DRIs for that protein bar specifically, right? That is just not feasible and not accessible to do for every food. So people came up with the daily values, DVs for short. And these daily values are the generic standards that are on food labels. So if you turn around the packaged food that you are eating and you see that label, that is called the daily values. And this is just a suggested guideline that will theoretically fit everyone in the public. However, this is theoretical, right? So it will not fit every person. For example, I think the individuals who are possibly younger than a certain age or older than a certain age or are pregnant will not necessarily fit in these daily values standards for a specific food. So always good to check with a physician or a provider before ingesting foods that may be risky. Okay, then we moved on to digestion, right? Once we eat the food, then what happens? Here you have a good schematic of the digestive tract, and I would say just an overview of the digestive tract. Know that you have the canal, right? The digestive canal. So you have through your mouth, the esophagus, then into the stomach, then into the small intestine, right? Goes around, then the large intestine, then rectum. So that is the digestive tract. However, around that, you have different accessory organs. For example, salivary glands in our mouth, or the pancreas, or the gallbladder. So for you to have a better understanding of all of the different functions of all the organs, I would definitely suggest going through this list right here and just reading to understand what happens at each step that the bolus of food travels through. Okay? Just, go over it. Could be easy points on an exam. I won't go over it because I'll just be reading it, but go ahead and stop and read this for sure. Here I've summarized some important points that Dr. Hampton talked about, so let's go over it. So the first place that the food goes to, right, the mouth. In the mouth, we are able to macerate the food, and macerate means to chew, right, and break down physically. We are also able to digest some carbohydrates through the enzymes that are produced by our salivary glands. The salivary glands also produce saliva, which allows for the lubrication and softening of the food in our mouth. And I believe the saliva also contains antimicrobial molecules and organelles. I think they're called lysozymes. Don't quote me on that. But for sure something antimicrobacterial to be able to fight off any germs that we may have eaten with the food. And then along with that, our tongue has many taste receptors, right? The 5 that we are focusing on in this class are sweet, sour, salty, umami, and bitter. And an important part about the taste receptors would be that every taste bud has different types of the taste receptors, right, within one taste bud. So that gives us a really cohesive understanding of what we are tasting. Okay. Then after the food, or now a bolus, which is what the food is called now, a bolus. I'll write it here, bolus. The bolus travels into the esophagus. And the esophagus is just a muscular tube that transfers the food from the mouth to the stomach. The functions of the stomach. So once we get to the stomach, the bolus of food is turned into chyme, And chyme is just the solubilized and digested version of the bolus, right? The ball of food that came into the stomach. And this happens through digesting some of the protein and mixing with some gastric juices. The exact functions that the stomach has is to release pepsinogen. Pepsinogen is the inactive form of the enzyme pepsin, and pepsin is an enzyme that breaks down protein. The stomach also reduces mucus, or releases mucus, and most importantly, it releases HCl, hydrochloric acid. Hydrochloric acid does a few things. It denatures proteins, right? Unfolds proteins, allows them to be digested further on. It hydrolyzes fats, breaks down fats, and it also kills microbes, right? So if the microbes that went past through this antimicrobial stuff in the mouth, then the HCl is definitely going to get them in the stomach. Okay? And HCl is also responsible for turning the pepsinogen into its active form, pepsin. Okay. This is taken from the lecture. So gastric pits are the structures in the wall of the stomach that release the HCl acid and the pepsinogen. The parietal cells release HCl. This may be incorrect now that I'm thinking about it. This may be incorrect. I'll check and possibly make an announcement. But I believe gastric pits only release pepsinogen. You can also look that up because I'm unsure. But parietal cells release the HCl. The chief cells are... Oh, I see. Okay, sorry about that. So gastric pits are just the pit of the stomach wall, right? And then in the gastric pits, we have parietal cells and chief cells and surface mucus cells. There you go. So yes, so the parietal cells release HCl, chief cells release the pepsinogen, which becomes pepsin later on. And the surface mucus cells produce mucus, which protects the stomach lining from the acid that it is producing. And that's an important part, right? Because if you think about it, HCl is actually, it has a very low pH. It has a pH of like 2, right? Or less than 2, meaning that it could be very damaging to tissue, to human tissue. So how is HCl released from parietal cells in a way that it doesn't eat through the parietal cell itself? Well, it happens through this mechanism right here. And we definitely didn't cover this in great detail, but I'll go ahead and cover it so that there's a better understanding for you as the student, but you definitely don't need to know the details for this class. So again, to get HCl into the lumen of the stomach, so inside the stomach, the parietal cell is going to produce the HCl, right? However, this HCl must be produced in a way to not be produced inside the parietal cell because it will destroy the parietal cell because it's so destructive, it's so acidic. So this mechanism is made in a way where the protons, the H+, and the chloride ions, Cl-, are pumped into the lumen of the stomach separate from one another. And then once they are pumped out of the parietal cell, separate from one another, then in the stomach, they join together to form HCl. Because in the stomach, we have this thick layer of mucus. So once it is in the stomach, it won't destroy the stomach. Okay, but if it was in here, then it would destroy the parietal cell. So how does this happen? Inside the parietal cell, we have carbon dioxide, which joins with a molecule of water. And through the help of the enzyme carbonic acid, it forms carbonic anhydrase, it forms carbonic acid. The carbonic acid then easily dissociates into a proton and bicarbonate, remember that bicarbonate is an important buffer in our system. So the bicarbonate just gets pumped out into the blood. Okay, because this is negatively charged and it just went out of the parietal cell, the cell wants to keep its charges equal, so it pumps in a negatively charged chloride back in to keep the negative charges equal in the cell. Now that chloride is in, it can travel through a chloride channel and it can just swim out into the lumen of the stomach. And up here, all these protons that are left here are just going to go through a proton pump and they're going to be pumped out into the lumen as well. And now we have the proton and the chloride out into the lumen by themselves, and they join together to form HCl inside the stomach. And that's how you get HCl in the stomach without actually producing it inside here and destroying the parietal cell. So hopefully that made sense. Again, you don't need to know those details. Just I gave you that background to hopefully give you a better understanding of what's going on. Let's see. And then another thing that I wanted to mention is the sphincters of the stomach. So we have the lower esophageal sphincter or the cardiac sphincter. It has two names. And then this is the pyloric sphincter right here. And sphincters, you can think of them as just, circular muscles around this area. And they just contract and they close up the stomach so that when the stomach is contracting and churning in all different directions to make up the chyme and mix up the gastric juices in the food, nothing actually goes up the esophagus or goes down here prematurely. Okay? And this is it for lecture 2. I will go ahead and make the videos for lecture 3 and 4 and move on. But if you have any questions, my name is Lilith. I'll write it right here. You can email me. My information can be found in the IATA resources module at the very bottom of Canvas. And I teach section AO2. So you can find my email address and everything there if you have any questions. Alrighty, thank you, and I'll see you when I see you.